Howdy, my name is Catherine Danmaigona Clement, and I will be sharing my research on fusarium wilt of cotton. Texas is the United States' highest producer of cotton, producing 39% of the U.S. cotton. However, fusarium wilt threatens the production of upland cotton from the recent expansion of this disease from California to Texas. Symptoms of this disease include up to 80% yield loss, early seedling death, wilting, and dark brown vascular discoloration. It is important to identify resistant sources in order to solve the problem of FOV, and it is also important to understand the nature of this pathogen. Our objective was to assemble this genome and understand if this genome do contain accessory chromosome, and we also set out to optimize an efficient protocol to screen for resistance to this pathogen. Our method include whole genome long-read sequencing and polishing the genome with short-read Illumina sequencing of 4 field isolate, which includes race 1, race 4, and 2 Texas strains. Our results show that we have optimized an efficient and rapid protocol that can help plant breeders eliminate 90% of their susceptible materials before sending them to the field for further testing. This will save plant breeders time and resources while trying to identify resistant sources to this devastating disease. This is the cycles plot showing the summary of our assembled genome, and I'll be showing 4 things with this plot. Number one is the fact that the FOV genome has 2 types of chromosome. The core chromosome, which is responsible for the normal function of the pathogen, and the accessory chromosome, which is responsible for the pathogen adaptation, virulence, duplication, and recombination. As you can see, this is a cycles plot showing 2 genomes. This is race 1 genome, which is much smaller than the race 4. And you can see the accessory or the race-specific region of race 4 is much bigger than the accessory or race-specific region of race 1. Another thing you should take note of is the fact that these accessory regions are more enriched in transposon as seen by the dark red color and the dark pink, which shows the repetitive sequence and the effectors. So these accessory regions are much more enriched in transposons, effectors, GC content, and DNA retransposon. This makes this region very prone to recombination and speed up the pathogen evolution and makes this pathogen much more devastated. We also observe a lot of gene duplication only confined within this accessory race-specific region, where you can see 1 copy gene in race 1 have to have up to 4 or 5 copies in race 4. Just by way of recap, we have estimated and optimized an efficient protocol to screen for resistance, and we have detected accessory chromosomes that might be important in the virulence and spread of this pathogen. This will help us in containing this pathogen from spreading into other southern states. Thank you for attention.